19, 20, 23, T-cell markers are tiny. CD3, CD4, CD5. There are also some markers like plasma cell markers, which I am not going to discuss. But this is the algorithm of lymphoma diagnosis and immunohistopathology and immunohistochemistry. So, for the breast is concerned, we have got common markers like ERPR, these are hormone sensitive markers, HER2, these are sensitive for targeted therapy, these indicate aggressiveness. So, by a permutation and combination of hormone sensitive markers ERPR and targeted therapy marker or the aggressive marker HER2, breast pathologists and the oncologists they have divided into four groups: luminal A, luminal B, enriched, and triple negative. If it is luminal A, it is only the hormone receptors are sensitive, HER2 is negative. If it is luminal B, both are positive and it indicates both hormone sensitive and targeted therapy sensitive. If only HER2 is positive, hormone sensitive receptors are negative, it indicates only targeted therapy, no response to hormone. And if both all three are negative, triple negative, then it is a candidate for chemotherapy or sometimes immunotherapy. Regarding the lung cancer, lung markers, it is very important to differentiate different types of lung cancer. Nowadays, lung cancers are classified into small cell and non-small cells, but our conventional classification can be held. That is squamous cell carcinoma, adenocarcinoma, and small cell carcinoma. Squamous cell carcinoma, CK7, CK6, and P 40 to 63. These are the markers for squamous cell carcinoma. For adenocarcinoma, it's CTF1, then it is CK7, and it is MAPSIN8. And for small cell carcinoma, synaptophysin, bromogranate, and CD56. Sometimes there are metastatic carcinoma also, depending on the immunohistological profile, you can search whether it is sarcoma marker or it is epithelial marker or it is lymphoma marker. Depending on what that we have definition. So this is about the applications of IHC. It locates the tumor origin, classifies the tumor, whether it's lymphoma, carcinoma, sarcoma. Most importantly, they help in unknown primary detection and tumors of uncertain histogenesis. The limitations in histochemistry is that many markers are expressed from different tissues simultaneously. So, a judicious selection of immunohistochemistry panel and clinical features, imaging features, and histopharmacology is necessary to pinpoint particular diagnosis of a cancer. Now, there is a term called morphometry. It is a computer-assisted quantitative geometric analysis of the cells, the tissues, the nucleus, and the nucleoli. It gives a total geometric assessment of the cell as a whole. It may be 2D images, it may be 3D images, and depending on that, we can assess whether a given cluster of cells is going to be malignant. If malignant, its aggressiveness, its activity, and its margin status. Now, there is a note on tumor markers. Tumor markers are products which are liberated by cancer cells or by normal cells in response to the cancerous tissue. What are the? They may be different biological nature. I remember with this mnemonic, there is a whole B. They may be hormones like testosterone, oncofetal products like CE and AFP. There may be line specific, for example, prostate cancer or immunoglobulins like multiple myeloma. There may be DNA markers like EGFR or BRAF mutated. There may be mucins and other glycoproteins like CA, 185, CA 15.5, or CA 15.3, or there may be enzymes. Now, most important role of tumor markers is apart from diagnosis, monitoring treatment and detecting their recurrence. Now, I come to a very important topic, that is flow cytometry. What happens here? Flow cytometry is a method by which a cell's character, single cell or a cluster of cells in suspension, their physical features, physical characters and chemical characters are detected. Thereby we can detect whether it is malignant or benign, particularly useful for immunophenotyping and hematolymphoid depletion. It has got three systems, three components. The cells in a suspension, which is stacked with immunofluorescent antibodies, they are passed through a beam of laser and immunofluorescence, that is fluorescence scattered particle immunofluorescence scattered light is liberated. And that light energy is converted to an electronic energy and detected by an instrument called photomultiplier. So this is very important in hematology and particularly our hematolymphoid neoplasia. Next we come to the molecular biology, which is very common, very popular to detect different translocations and chromosomal anomalies in the tumors. We know I am not going into detail of the principle of polymerase chain reaction, but it helps to detect the chromosomal anomalies. Also, it detects the associated infections, particularly in diagnosis of oncogenic viruses and some oncogenic bacteria like Helicobacter pylori. Next, we come to fish study, which is very important in diagnosing some of the translocations in solid tumors like synovial sarcoma, Ewing sarcoma, and mantle cell lymphoma. It is single double-stranded DNA is denatured into single DNA, and the fluorescence potent immunofluorescence potent probe is hybridized, it is washed, and it is seen under microscope to detect a particular translocation in a particular DNA segment. Now, how oncopathologists can prognosticate